Welcome, everyone. Thank you for joining us at Navigating Spatial Biology. Take that next step with Leica Biosystems. Our next presentation for today is introducing new RNA protein co-detection assays, presented by Madhura Nayak and Anushka Dikshit. My name is Marie, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To ask a question, click on the Q&A box located on the bottom of your screen, type your questions, and hit Submit. The speaker will answer your questions following the presentation. If you have trouble seeing or hearing the presentation, please click on the Q&A box to let us know that you're experiencing a problem. Please join me in welcoming Madhura and Anushka. For a complete biography on our speakers, visit the Biography tab on the left. Madhura and Anushka, over to you.
Thank you, Madhura. For those of you who are not familiar with the RNA Scope technology, it's an in situ hybridization platform designed for detecting specific RNA biomarkers. On this slide, you can see I have described the three major assays falling under the RNA Scope technology umbrella for detecting RNA biomarkers. The first is the RNA Scope assay, which is specifically for targets that are over 300 nucleotides in length. This assay is available as a chromogenic or fluorescent assay and in different configurations of automated or manual assays. The second is our BaseScope assay, which is a specialized assay designed for targets that are between 50 to 300 nucleotides in length. These include your small molecules like splice variants, exon-exon junctions, highly homologous sequences, and also point mutations. This is again either a singleplex or duplex assay available in automated or manual platforms. And finally, our newest assay is the microRNA scope assay, which, as the name suggests, is for targets even smaller than 50 nucleotides. And these are for targets like microRNAs, ASOs, or siRNAs. And currently, this is available as a singleplex assay in both automated and manual platform. In addition to our RNA detection assays, we've also launched a DNA scope assay this summer, which allows you to visualize DNA biomarkers. So why is it important for us to look at RNA biomarkers with a highly precise tool like RNA Scope? In the last few years, we have understood the importance of spatial resolutions since it allows us to study highly heterogeneous tissues like the embryo or the brain or tumor, wherein you have multiple cell types that are constantly interacting with each other, communicating, and determining the function of the target tissue. Tools like RNA Scope are highly specific and allow you to visualize your target RNA biomarker or target of interest with high sensitivity and specificity at single cell and single molecule resolution, and thereby allowing you to elucidate the heterogeneity of these tissues and study the cellular interactions. While it's important to study spatial expression of RNA biomarkers, it's even more important to get a multi-omic analysis of your complex tissues, thereby understanding the dynamic interactions happening between different cell types. And realizing this, we have enabled combining our RNA Scope ISH assays with immunofluorescence or immunohistochemistry, thereby allowing detection of your RNA biomarkers in combination with target protein biomarkers. This allows us to visualize target gene expression in target cell It allows us to identify cellular source of secreted factors like chemokines, cytokines, growth factors. It also allows us to get a better spatial visualization of target cells by marking the cell border using a specific antibody and visualizing gene expression with single cell resolution using RNA Scope probes. And therefore, by combining RNA Scope with either immunohistochemistry or immunofluorescence, we can leverage the best of proteomic and transcriptomic approaches with high resolution and specificity. In the next few slides, let's look at a few examples of how RNA Scope can be combined with immunohistochemistry or immunofluorescence for a range of different applications, starting with examining cell type specific gene expression. In this particular example, they're studying the expression and role of PRDM12, which is a transcriptional regulator, and its role in development of pain sensing neurons. So for this example, they used a specific PRDM12 RNA Scope probe and combined it with 3 different antibodies to study which of the subtype of these neurons were expressing PRDM12 and therefore responsible for the development of these pain sensing neurons. We observed that when they combined it with TRKA, they saw a very strong co-expression of PRDM12 with TRKA, while on the other hand, the correlation was not as strong when they combined it with either TRKC or TRKB antibodies. And this told them that there was the strong correlation of PRDM12 with TRKA shows that PRDM12 is expressed in almost all of the nociceptive neurons, but the expression was not as strong in proprioceptive or mechanoceptive neurons. This revealed that the presence of the PRDM12 transcription regulator in these TRKA positive neurons suggested that it was essential for the development of these pain sensing neurons. In this next example, they have used the Base-Scope assay and Base-Scope assay with combination with IHC to validate single nuclear RNA sequencing data. Here, they are studying multiple sclerosis and trying to understand the differential cell populations in MS brain versus a normal brain. They were able to obtain postmortem MS brain and compare it to a normal brain and study expression of different RNA markers. Here you can see that they have identified different sets of neuronal and non-neuronal populations in the brain, and they see some differential expression between wild type and MS brain. So they used a few markers specific to oligodendrocyte populations and studied their expression in this control brain versus brain with MS. And here you can see with the Base-Scope assay, the dual Base-Scope assay, they were able to identify these different populations of oligodendrocytes by using subpopulation specific markers. Next, they wanted to also identify what are the different populations of cells that are potentially present in a control brain versus MS brain. And so they compared a MS lesion and visualized what they're already seen as a part of the single nuclear-seq RNA sequencing. That is, that they identified a hybrid cell called oligodendroglial cell having characteristics of both the oligodendrocyte and the microglia, and therefore expressed markers belonging to both cell types. And they confirmed this by doing dual Base-Scope IHC by using CD74 a microglial marker and olig-1,2 oligodendrocyte marker, and showed that these two markers, which are specific to different types of cells, were actually co-localizing in this hybrid oligodendroglial cell. And this cell was present in abundance in the MS lesion, suggesting that this hybrid cell population could have something to do with the MS pathology and might contribute to certain pathological features associated with MS. In the next example, let's look at how RNA Scope can be used in combination with immunofluorescence to identify soluble factors and the cell of origin secreting these soluble factors. This is an example where the researchers were studying pancreatic cancer and trying to identify whether LIF, which is leukocyte inhibitory factor, had a role to play in progression of pancreatic cancers. They identified that in patients with pancreatic cancer, there are high levels of circulating LIF in their blood compared to normal individuals. Similarly, they also identified that presence of LIF mRNA correlates to decreased disease-free survival in these patients. What they wanted to identify is how this LIF was actually contributing in growth and progression of pancreatic cancer. So they used RNA Scope in combination with immunofluorescence to study the expression of LIF and identify how it might impact growth of pancreatic cancer cells. In this image, you can see that they combined immunofluorescence for keratin 19 with that of LIF RNA Scope probe and PTPRC or CD45 RNA Scope probe. And you can see that LIF and PTPRC were present in the stromal region of this tumor, while keratin 19 expression was, as you can imagine, specifically localized to the tumor cells, suggesting that LIF is expressed by potentially by the pancreatic stellate cells in the stroma and was exerting its impact in a more paracrine fashion to promote growth of pancreatic cancer cells. Let's look at another example of how RNA Scope was combined with IHC to identify a specific population of immune cells. In this paper that was published in Nature Medicine, these researchers were treating a melanoma patient with anti-PD-1 therapy, and this patient unfortunately reacted adversely and led to encephalitis in this patient, which was ultimately fatal. But these researchers are interested in understanding which immune cell populations were responsible for inducing encephalitis in this patient. So they performed TCR sequencing on this inflamed brain tissue and identified sequence that was most frequently occurring and designed a specific base scope probe against this TCR sequence. They then localized this sequence with CD4 and CD8 antibodies to identify which type of T cells were present in this encephalized brain. And surprisingly, they were able to show that this TCR sequence co-localized with CD4 positive T cells and not with CD8 positive T cells, suggesting that it was the CD4 memory T cell population that was responsible for inducing this encephalitis triggered by PD-1 treatment. So this was a very important finding because it's important to understand the type of adverse events that can happen due to immunotherapies and the type of cell populations responsible for induction of these adverse events. In this next example, they want to identify regulatory mechanisms of PD-L1 mRNA. Although checkpoint therapies have been extremely successful for a number of malignancies, we don't yet completely understand how these immune checkpoint receptors and markers are regulated at the mRNA level. Franchini and colleagues identified a post-transcriptional regulatory mechanism by which PD-1 mRNA was regulated in T cells. They identified that PD-1 mRNA is transported via microtubules and stress granules before they are translated in the cytoplasm. And to demonstrate that, they utilized RNA Scope in combination with immunofluorescence and showed us in this image, as you can see, when the cells were untreated, you can see nice tubulin staining and PD1 staining, as well as presence of PD1 protein in yellow, and a nice co-localization of the mRNA and protein, as you would imagine, in presence of intact tubulin. But when the cells were treated with a microtubule inhibitor, you can see that the expression of mRNA is present as expected. The tubulin marker is not present anymore. And also, the PD1 protein is very low or almost negligible in expression, suggesting that the mRNA was not successfully transported to the cytoplasm and therefore did not get translated efficiently. Furthermore, they also wanted to show that this was happening via the stress granules and used stress granule specific marker G3BP1 antibody and combined it with the PD1 transcript probe, showing nice co-localization of the stress granule biomarker, stress granule marker with that of PD1 mRNA, suggesting that the transport was indeed via stress granules and microtubules, and is important regulatory mechanism of the PD1 mRNA. In this next example, we'll see how this workflow can be successfully used to identify certain biomarkers and drug targets. In this particular example, these researchers are studying the Alzheimer's disease model, and although we have made significant advances in understanding Alzheimer's disease, we still don't understand the association of senescent cells with that of certain physiological characteristics associated with Alzheimer's disease, including cognitive impairment. And so these researchers wanted to demonstrate the presence of these senescent cells in the amyloid beta plaques found in the Alzheimer's brain. And so they used RNA Scope P16 probe in combination with the AB antibody to demonstrate that P16 positive cells were indeed present in these amyloid beta plaques in this mouse model. They went a step further to show that with 3D microscopy and imaging, they were able to study the 3D structure of the plaque and showcase that the cells were dispersed throughout the plaques. The senescent cells at P16 were present throughout the plaques. Next, they also wanted to show that by reducing senescence in these animal models, they could potentially suppress neuropathology associated with Alzheimer's disease and other degenerative conditions. And so for that, they treated these Alzheimer's disease mice with 2 senolytic compounds that are FDA approved. That is 2 compounds that would prevent senescence. And when they treated these mice with these senolytic compounds, you can see compared to the vehicle, the treated sample had very low to no expression of P16, suggesting that the senescence was significantly reduced in these treated models. Next, we're going to look at an example of how this technique is specific enough to look at the spatial organization of a tissue and identify niche specific expression of RNA biomarkers. Here in this example, they are studying the role of CD19 positive fibroblasts in differentiation and homeostasis of the intestinal stem cells. As you all know, intestinal stem cells are important as they renew the intestinal epithelium, which is important for a healthy gut function. So what they did was they combined CD19 specific RNA Scope probe with that of various different antibodies for specific cell type specific markers. So in this first image, they've combined CD90 with GP38, which is a fibroblast specific marker. And as expected, there is nice co-localization of this GP38 with the CD9 positive cells and this intestinal crypts. They also combined CD90 with BRDU, which is an indicator of proliferating cells. And by using BRDU antibody, they were showing that CD19 was in proximity to these epithelial cells that were proliferating. And finally, they were able to show that CD19 was also proximal to LGR5 positive cells in the crypt, which are potentially your intestinal stem cells. And so how does CD19 positive fibroblasts actually exert their effect on these stem cells and regulate differentiation of these epithelial cells? They were able to show that by expressing or by secreting soluble factor CMA3, the fibroblasts, the CD90 positive fibroblasts, are actually able to exert this impact on the epithelial cells and promote differentiation and homeostasis of these stem cells. And they were also able to show that NPR2, which is a marker for, a receptor for CMA3, was also found on the epithelial cells of these intestines. So with this workflow of combining RNA Scope with immunofluorescence, they were able to establish the role of these CD90 positive fibroblasts and demonstrate how they were able to regulate differentiation of these stem cells in the intestinal crypts. Finally, the last example, I want to show you how, in this last example, let's study how these researchers used RNA Scope in combination with immunofluorescence to study infection by the Rift Valley virus. The Rift Valley fever virus is a livestock disease that can potentially have zoonotic transmission to humans. We are not really clear on how this might actually impact pathology in the brain if it impacts the humans. And so these researchers studied the infection of this virus by using a rat model. They utilized specific RNA Scope probe to the viral RNA and combined that with IBA1 positive antibody to study infiltration of immune cells post-infection in this rat brain. As you can see, compared to the uninfected image, there is increased presence of the viral mRNA and immune infiltration post-infection day from 1 to day 7, suggesting that there can be potentially significant neuroinflammation related to this viral infection. With that, I would like to transfer the platform back to Madhura, and thank you for your attention today.
Thank you, Anushka, for that excellent overview of the need to detect RNA in protein and showing us some great examples of applicable research questions addressed with this. And with that, I would like to introduce you to our reagents that enable these applications. The new reagents in the form of the RNA protein co-detection ancillary kit, when used with the new workflow, will allow researchers to simultaneously examine cell type specific gene expression and identify cellular sources of secreted proteins. Some of you may have already tested certain antibodies and found them incompatible with our RNA Scope and base scope assays. Our aim with this new assay is to enable more antibodies to be compatible with our assays, and we will see later some examples of key antibodies that were previously incompatible are now compatible with this new workflow. The new assays essentially contain 3 parts. The new RNA protein co-detection ancillary kit, which includes the co-detection blocker, co-detection target retrieval solution, and the co-detection antibody diluent, which is also available as a standalone item. This kit has been validated with our chromogenic red and multiplex fluorescent assays on both the manual and the Leica platforms, and also has been validated with our base scope assays. Along with this kit, we would also like to introduce you to the new protocol where the key primary antibody step is now performed right after the target retrieval step, following which we go on to perform the rest of the RNA ISH steps. We then follow this up with a secondary antibody to detect the protein of interest on the same section, and this workflow has been designed to be completed within 2 days, with the primary antibody incubation lasting overnight as the breakpoint. With this new integrated workflow approach, we have now enabled more antibodies to be compatible with the RNA protein co-detection and reduced the need for extensive screening for compatible antibody clones, resulting in faster time to results. For those of you who may have tested the sequential workflow before, these new reagents and workflows support equivalent or better performance of antibodies and also supports equivalent sensitivity to ISH-only detection and more faithfully retains specificity of the antibody in the IHC alone. In this slide, I'm showing what a signal from an IHC-only run looks like in the left panel. And in the final panel on the right, with our integrated workflow and the new pre-treatment reagents, we can now rescue the IHC signal, and this in a sense makes it easier to combine your antibody of choice with RNA ISH. To enable this compatibility, our team has developed some key reagents, the first of which is the target retrieval solution. The target retrieval with the current RNA Scope and base scope assays has been thoroughly validated and is intended for use with the RNA ISH assays alone. However, in some instances, these may have impacted the protein detection. This new co-detection target retrieval reagent has been formulated for improved protein detection while maintaining equivalent ISH conditions or ISH detection. The second reagent is the co-detection antibody diluent. In our experience, certain commercially available antibody diluents have negatively impacted RNA ISH staining, as can be seen from the image in the middle panel on this slide. This drove us to develop the new diluent which maintains both RNA ISH and IHC staining. Lastly, to ensure no cross-detection between the two chemistries, a blocker is needed, which is only required for the manual chromogenic assays. The main difference between the previous workflow and the new reagents in the workflow is that with the sequential workflow, extensive prior optimization was needed to ensure antibody compatibility, whereas now a pre-validated list is no longer needed, and most IHC compatible antibodies can now be combined with the RNA ISH assay. An example is the CD8 antibody shown here in the left panel with standard IHC, in the middle panel with ISH and IHC without the new reagents in the protocol where we lose the signal, and in the right panel we are now able to visualize both signals with the new co-detection reagents. And with that, I would like to do a quick recap. If you're running our manual assay, you would need the ancillary kit. And if you're running this on the Leica platform or the Leica Bond RX instrument, you only need the antibody diluent. The assays compatible on the Leica Bond RX are the 2.5 assay, the red assay, the LS multiplex fluorescent assay, as well as the base scope LS red assay. In this slide, we create a reference for the reagents you would need for each protocol that we have validated with our assays. As an example, if you're running our manual assay, red assay, with the new reagents and workflow, you would need all three reagents provided in the ancillary kit. Please reach out to our excellent support team to get help with getting started with these new assays. Now, I would like to take you through some examples of staining generated with the new reagents and each of our validated assays with IHC antibodies. In the first example here, we show some data with CD4, CD5, and FOXP3. CD markers are cell surface markers which are very useful for the identification and characterization of leukocytes and the different subpopulations of leukocytes. CD marker-specific antibodies have been widely used for cell sorting, identification, and cancer diagnosis. FOXP3 is a protein involved in immune system responses and known to be a master regulator of the regulatory pathway in the development and function of regulatory T cells. These represent fairly standard proteins utilized in oncology research, and shown here, combined with RNA ISH, can be used to get more data out of precious samples. The samples shown here are tonsil, head and neck cancer, and as you can see from the panel on the right, both RNA ISH and IHC signal is maintained with the new workflow and reagent with our Leica red assay. Another example of staining here, the protein detection is performed using our green chromogen, giving a great contrast on tissues where an alternate to the brown DAB is preferred, perhaps due to tissue artifacts. Shown here are examples of CD3, CD8, and Ki67 antibodies in the left panel as control IHC alone. In the middle panel, combined with RNA ISH without the new reagents and workflow. In the right panel, we observed optimal signal detection with both the RNA and protein. This run was performed with our manual assay. In this slide, we show compatibility of these antibodies and workflow with our Bioscope assays on the Leica platform. Moving on to fluorescent ISH and IF, these new reagents and workflow have also been validated with our fluorescent, multiplex fluorescent assays, where we use the TSA detection system to visualize the signals. Shown as an example here are the CD3 and CD8 antibodies on lung cancer tissue with control probe PPIB for RNA ISH. The antibody signal is in white and the RNA scope signal is in red. Here is an example of 3-plex ISH with Ki67 antibody with this new reagents and workflow, a key example of how you can get much more data with more targets and detect both RNA and protein on the same slide. Here we're highlighting some antibodies, specifically CD markers that may have previously been incompatible in combining with RNA ISH and required extensive screening and optimization. And lastly, here are some more examples of CD markers detected with the brown or the green chromogen along with the red ISH signal. And with all that data, I would like to conclude and summarize today's presentation. In today's session, we have shown some key examples of how multiomics profiling with single cell resolution can be achieved with the new reagents and workflow. With this new ancillary kit, you can now incorporate RNA ISH into your existing IHC workflow to gain deeper insights and preserve precious samples. This new reagent and workflow have been extensively validated with the red chromogenic and multiplex fluorescent assays on manual and the Leica platforms. And lastly, we hope that with this product and key research examples we have shown in this deck, you're now able to utilize the power of two technologies, IHC and RNA ISH, to answer your research questions. With that, we will now address some questions that we have coming through. Thank you.
Thank you, Madhura and Anushka, for that informative presentation. Just a quick reminder to our audience on how to submit questions. Simply type them into the Q&A box and click on the send button. We will answer as many questions as we have time for. Our first question is, does the new co-detection workflow and reagents impact tissue morphology or the RNA signal?
Thanks, Maria. This is Madhura, and I can probably take that question. So the short answer is no. We've actually really formulated or reformulated some of the reagents, as you saw in our presentation today. We have a new target retrieval buffer. We also have taken great care to provide specific, antibody diluents, in which we recommend that you dilute your antibody of choice. And that, in fact, was done in order to maintain both the tissue morphology and also the RNA signal. as we've shown through the various examples in the presentation today, we've shown that the signal intensity of both the RNA and actually the protein is very well maintained when utilized with this new co-detection workflow in conjunction with the new reagents that we've launched last year.
Great, thank you. Next question. Can this workflow be used to identify a clonal population of T cells?
I can take that one, Marie. Thank you so much for that question. And this is really important because for IO researchers who are trying to develop new immunotherapies, it's important to identify specific population of T cells. And with our base scope assay in combination with co-detection, you can detect specific CDR3 sequences on T cell receptors and then use antibodies against CD4 or CD8 to identify that specific population of T cells. So with combining our co-detection assays with our base scope assays, you will be able to identify specific clonal populations of T cells.
Great, thank you. And we have time for one more question. Can the new Co-D workflow be combined with fluorescent assays?
Yeah, that's a great question, and the answer is yes, absolutely. In fact, if you were to utilize the fluorescent assays that we currently have available on the Leica platform, you can actually flex higher on the RNA target. So with our fluorescent assays, you have the capacity to actually combine up to 4 RNA targets of interest. And then you can additionally, if you have the filter set, you can then add in your protein target of interest as well. So typically, if you have a standard 4 filter set, what you could do is you could combine 3 RNA targets, and you could utilize the 4th channel for your antibody of interest, which will target your protein of interest. And that way you'll have a 3+ RNA assay and a 1-plex combined with a protein target marker of interest. So absolutely, yes, you can combine this new workflow with the fluorescent assays, and we have again demonstrated that through some of the very beautiful images and data sets that we've just presented in the webinar today.
Well, great. Thank you so much. Do either of you have any final comments for our audience? All right, well then a big thank you to Madhura and Anushka, and thank you to our audience for joining us today and for their interesting questions. We will address questions we did not have time for today and those submitted during our on-demand period via the contact information you provided at the time of registration. This session webcast will be available on demand after the event. That's all for this session. Thank you for joining us, and we hope you have enjoyed this presentation. Bye for now.
Thank you.
Thank you.